Hi, everyone. Welcome to today's HERN webinar entitled Bridging the Gap, Pancreas Tissue Slices from Organ and Tissue Donors for the Study of Diabetes Pathogenesis. My name is John Cadiz, and I'm going to introduce our speakers today, Dr. Stephan Speier and Dr. Alejandro Caicedo. So in today's webinar, for those of you that may be joining us for the first time, I want to welcome you, and we'll give you a brief overview of what HERN is, and then we'll jump right into the science. And we'll answer the question, what is a pancreas tissue slice? Some of you, I'm certain, are familiar with tissue slices, and you've known about it. Others of you may not. And then we'll look at how these tissue slices can be used, what's been discovered with these tissue slices, and then some of the limitations and considerations when you're using a tissue slice. And then I hope at the end we can get a good Q&A session, and I'll encourage you to ask the speakers directly your questions, but if you'd like to put them in the chat at the end, I'll moderate the chat session, and I'll queue up those questions to the speakers as well, so we'll make sure that we don't miss anything. So just a little bit about what HERN is. HERN is a very large network that began in 2014, and it's made up of several different groups, as you can see pictured here, and each of these groups focus on a particular theme. So there is a consortium, for example, on beta cell death and survival, another one on autoimmune interactions, and so on. As you go through the circle here, each of these groups will, again, be focused on something, and they'll be asking different questions, but tying that theme together. Today, we're going to hear from the Human Pancreas Analysis Consortium. And again, Drs. Caicedo and Speyer are members of HPAC, where together with Dr. Marcela Brisova, they lead a project looking at human pancreas tissue slices and examining postnatally how the endocrine, vascular, and immune components work together to form an islet. So they've been interested in this for some time. In all, HERN has over 200 investigators in 9 different countries, 80 different institutions. In fact, Dr. Speyer is in Germany now. Dr. Caicedo is at the University of Miami. So this is sort of a worldwide initiative. And if you're curious about what these groups are doing, HERN publishes annual reports every single year that you can get on the website. The most recent report is in progress, and it should be out in a few months. There's also publications that come out and that we keep track of those, so you can see all of those. Speaking of the website, we just released, and some of you may have noticed, a new HERN website where all of this information can be accessed. I'd like to draw your attention to the follow panel on the right-hand side, where you can Facebook, Twitter, LinkedIn, Instagram, but particularly the YouTube, where all of these recordings, our monthly talks, are recorded. We have a YouTube channel. You can get to it from the main website, and you'll see this and past presentations as well. But in case you're curious about the kinds of things that are coming out of HERN in terms of the resources, we also have a resource catalog where we gather data sets, cell lines, various different tools and discoveries that scientists are making within the HERN, and catalog those and make those available for others to use. And in today's webinar, we have 2 of the protocols that Dr. Speyer and Alejandro have prepared. They're available here. This work has also been published and accessible from the website as well. So with that brief introduction, I'm going to hand this off to Dr. Speyer, and as I do, I'm going to introduce him to you. Dr. Speyer is a professor of medicine, actually professor of pancreatic islet cell physiology as the Faculty of Medicine at the Technical University of Dresden in Germany. And Dr. Alejandro is a professor of medicine at the School of Medicine in the University of Miami, where he is interested in neurobiology and eyelid physiology. So both of these guys share an interest in physiology, and I suspect that's probably what brought them together on this project. So with that said, I'd like to turn it over to you, Dr. Speyer, and I look forward to your presentation and that of Dr. Caicedo.
Thank you, John. Do you see the slides? Is that okay?
Yeah, that's perfect. Thank you.
Okay, so thanks, John. Welcome, everybody, and thanks for the opportunity for us to present here. As already introduced by John, this is a joint endeavor by Alejandro Caicedo and myself, and we want to talk about pancreas tissue slices, and especially towards the end, focus more and more on the human part of this and why we think this is important to look in the human pancreas for the pathogenesis of diabetes. So we will split this up. I will start. I will tell you a little bit about the history of this and how we got actually to develop this platform, how this all started in mice, and then switch to human. And then Alejandro will take over and show you some examples, some exciting new unpublished discoveries. And at the end, I will try to put the platform a bit in context with other platforms that we use to study the human pancreas, and look also at limitations of this platform. So for those that are not working in eyelids, as this is a public webinar, I just wanted to shortly introduce why we're actually doing this. So most of you know we have the islets of Langerhans, which are endocrine cell clusters that are distributed throughout the pancreas. And these clusters are made of different cell types, and they secrete hormones. So we have beta, alpha, and delta cells, for example, and they secrete insulin, glucagon, and somatostatin. And they are responsible for glucose homeostasis and to keep us all in a normal glycemic range. So in some cases, this goes wrong. For example, as you know, in type 1 diabetes, there is an autoimmune attack. The patients have reduced number of islets and cells, and also reduced function and in the beta cells, and that leads to a hyperglycemic setting. And in type 2 diabetes, this is a little bit more complex because also other organs play a role in terms of increased insulin resistance. And in addition, the cells from the islets are dysfunctioning as well. So this is basically why we're interested in these various states and study the biology of islets and islet cells and want to understand how the physiology and the pathophysiology of islets work. And I started up, as many others, in using mice, because at the time when I started to study, there was not much human tissue around to look at. And we are still looking a lot in mice and learning a lot from mice and other animal models. But then we try to translate this more and more into the human setting. And this tissue slice approach is actually one that has helped us a lot to do so. So when we started, we were usually studying mice in a systemic way, so systemic metabolism. A few labs around the world were or are able to do a perfused pancreas, which then allows you to be a bit more focused. You don't have all other organs, all other systemic modulators in place. And but I guess most of us that studied islets at a functional level, where you're then using isolated islets or are using isolated islets from mice to look into the physiology and pathophysiology. And of course, you can further disperse the islets and look at a single cell level. And when I started, and we were using these techniques in the lab, and but then we had questions that we wanted to look at, and we realized the technologies that we have do not really allow us to do so. So we, for example, had cases where we looked at phenotypes where the morphology of islets was disrupted, and if we were trying to isolate the islets, we would not get islets that were really isolated. We later on wanted to look at infiltration of islets in autoimmune models, and most of the immune cells were also lost during the isolation process. And in that case, when we were not able at that time to do in vivo imaging. And of course, if you then look at modulatory factors of islets, this is difficult to do so if you isolate the islets out of the pancreas, because you will lose certain aspects of the vascularization and also, of course, innervation. Like if you would be interested in looking at the neurons, the complex, which usually sits on the outside of islets. So we had a question back then, and that was, how do we look at islets in a more intact environment so that we can address some of these questions? And back at the time, I was a PhD student in the lab of Mayan Rupnik, and we both looked a little bit younger at the time. And so he came up with the idea when he came back from a meeting, why not let's inject agarose into the bile duct of a mouse and harden the tissue. It's a low temperature gelling agarose. And we tried this, and after lots of tries, it actually worked. And so the tissue hardens up, and you're still at physiological temperatures during the whole process. And then you can extract the pancreas, and you can cut out blocks, and then you can actually slice this in various thicknesses as you wish, depending on what you want to study. We usually were around 150 to 80 microns, something like that. So what we ended up with at the time was something like this, and you can see this is basically living pancreatic tissue. You see the exocrine part around here. This is a dimension of about 5 times 5 millimeters. And what you see here are the islets. In this case, they are nicely aligned along a larger duct. And you can even see them through the stereo microscope without any staining or anything because of the differences, how they scatter the light. And if you zoom in a little bit and then look in transmitted light, you can see nice ellipsoid islets, which we found a lot in the slices here next to a duct and a vascular structure, and again surrounded by exocrine tissue. So when we had these slices, at the time we were actually a patch clamp lab. So what we did most was do a patch clamp. And we did a number of studies then in the beginning, looking at the electrical properties of cells. We saw that there are some differences in the physiological status of beta cells within in a slice in comparison to the other preparations that we looked before. And then we continued to start doing insulin release measurements from the slices. We did exocrine enzyme release in the slices to prove that also the exocrine part is still alive and functional. And we started to do live cell imaging in both parts of the pancreas. So, and then we were working on pancreas slices for some years. We had some publications. Other labs started working on slices, like Alejandro's lab, or the lab from Herbert Caicedo started also working on slices. And then when I got my own lab in Dresden, we had the idea to why not try to do this with human tissue. So we had access to human tissue, but we didn't have access to human islets. What we could get, because of a collaboration with Michele Solimena from the university and Jürgen Weitz from the clinic, we could get pieces of pancreas tissue from pancreatectomized patients that would undergo pancreatectomy because of a pancreatic or close to the pancreas related disease. And these pieces were too small to really isolate islets from it. And so what we did is we tried if we could do tissue slices on those. And after adjusting some parts, we were able to also get tissue slices from the human pancreas. And now we could actually study the human pancreas. So pieces of living human pancreas with islets inside the exocrine part. What you see here is also adipose tissue, which you see very frequently in the human pancreas. And shortly after that, and after presenting the technology at a meeting, I was approached by Alberto Pugliese, and we teamed up with NPOD, and together with Alejandro's lab and the lab of Alberto Pugliese, and people from NPOD, from Mark Atkinson's lab, We then set up the approach to slice tissue from organ donors that comes in EndPot. So first we established this in Miami, and then we transferred the technology to Gainesville. And so we managed all together to get the same approach going from tissue that we got from organ donors as we had from tissue donors from pancreatectomies. So two proof of concept studies that we did since then, which are published, which I'm just going to show you very fast because the exciting new data is going to come from Alejo's side. So you might have seen this one when we did the first study on the pancreatectomized patients. The fascinating thing for us was that we could phenotype these patients before the surgery. So we knew exactly their blood glucose levels, HbA1c, their glucose tolerance. We knew a lot more. So we could put them into categories of non-diabetic, impaired glucose tolerant, or type 2 diabetic. We actually had more categories like that, but for the publication, we left it to these three categories. And we asked a very simple question when we got tissue from the pancreas of these patients: why do they secrete or why do they have too little insulin in the blood? And we wanted to know, is it because they have too few cells, or is it because the cells are not working? And to cut a long story short, what we saw was already at the impaired glucose tolerance state, we saw changes in secretion, but no changes in mass. We saw increased basal and reduced first phase response, but the second phase was unchanged. And even in the diabetics that were for many years diabetics, in the tissue that we looked at, there was no change in beta cell mass, but a severe reduction. So this was in the case of type 2 diabetes. So in the case of type 1 diabetes, where we did the first study, again, I'm just going to go very shortly over this. This is published. What we did is we, together with EndPot, we got non-diabetic cases, and we got tissue from 4 type 1 diabetic organ donors with different durations of the disease. We did the slices, and then together with the labs in Gainesville, in Miami, and our lab, we then looked at the hormone secretion, the morphology in these slices, and at calcium imaging. And then we could put, for every case, we could make a detailed endocrine profile of the function and the morphology of these donor cases that we got, which then allowed us to get a bit more in detail information, what was going on in these individual cases of diabetes. So as I said, these were published studies, and I now would like to hand over to Alejandro, who's actually going to show you some exciting new data done with human tissue slices.
Okay. Hi, everybody. So my task is to illustrate with some examples the power of this technique. And the first example that I'm going to use is how we are using now these slices from type 1 diabetic donors to study certain endocrine cell dysfunction in type 1 diabetes. So the work that I'm going to present was performed by Julia Panzer, who used to be, who graduated with Stephan Speier in Dresden, is now a postdoc in my lab. And of course, we are all interested in understanding what leads to endocrine cell dysfunction in diabetes. Of course, in the case of the beta cell, probably it's just disappearing and being destroyed. But on the way there is also some residual function, and we are using both slices as well as isolated islets. And if you look, let me point with a laser. No, it's not here, just a laser point. Okay. Here, this is an example of a human slice, and you can see islets within the slice, and these are isolated human islets from diabetic donors. What you can see in the anatomical image is that in the case of the slices, you have many islets that do not contain any insulin or beta cells. while in most of the isolated human islets, you still have some remnants of the beta cells. But if you look at the secretion here and you compare the secretion from non-diabetic donors to the secretion from a type 1 diabetic donor, type 1 diabetic donors, you see that there's a very, very strong difference between the two. And this is also true for the secretion from isolated islets. But if you look at closer at the secretion from these islets, you see that there is some residual function. By no means I want to say that the isolated islets are worse than the human, the slices. But what is maybe true is that there is sort of a survival bias when you look at the islets. It's very, as Stephan said before, it's very difficult to isolate islets that are destroyed by either the immune system or by other reasons. So there may be an advantage in this sense by using the slices. So what we are interested in is in understanding the mechanisms that lead to defective glucose counter-regulation, to defective glucagon secretion, and hypoglycemia in type 1 diabetes. So this is a major problem. If it weren't for hypoglycemia, I think that type 1 diabetes could be treated very aggressively with insulin. But the problem, as you can see here, is that they are paradoxically prone to hypoglycemia when they inject insulin. And you can see one of the reasons is because these patients, they do not secrete glucagon in response to a lowering in glucose. So what can we do to study this here? We use pancreas slices, again, from NPOT. And this is glucagon secretion from slices. You can see that in red, the slices, the recordings from slices from type 1 diabetic donors. And you can see that the baseline level of glucagon secretion is higher than in the control non-diabetic donors. And this is plotted here in the middle. Here you can see a quantification from 4 different patients compared to 4 different non-diabetic donors. Now, but if you look closer at glucagon secretion and you show it as a fold change over baseline, you can see that the natural response that the alpha cells have to a lowering glucose is there in the slices from non-diabetic donors, but it completely disappears in the slices from diabetic donors. So this is a major change, and now we think that we have the tools to look at the mechanisms. Of course, glucagon secretion is difficult, so it's easier to look at some of the mechanisms if you do calcium imaging. So we are able to load these slices with calcium indicators, and then we can record calcium activity in the individual cells. This is a heat map that you see here, where we plot the response of the cell over time here, and you see the individual cells are the individual rows here. And the intensity of the response, the intensity of the increase in the fluorescence of the calcium indicator is presented in a pseudo-Carter scale here. And if you compare the response in non-diabetic donors, slices from non-diabetic donors, you can see that the lowering in glucose elicits a strong response in many, many cells here. In slices from type 1 diabetic donors, you can see that this response is much less, first of all, less frequent. And also, if the response is not as strong as the responses that you see in slices from non-diabetic donors. This is plotted on the right. And here, what I show is the comparison of the calcium response of these cells to the secretion that we see, the glucagon secretion, and it looks very similar. So we have a tool now, an in vitro tool, where we can look at mechanisms. I'm not going to go further into or deeper into the mechanisms because it will take the whole seminar, but this is just to tell you that we have the tools now to look at what are the mechanisms that are wrong in the alpha cells in type 1 diabetes. So, and a second example is a study that we started now already 2 or 3 years ago to look at the postnatal maturation of human eyelid function. This has been very difficult. Historically, it's very difficult to get samples from very young donors. So this is, of course, a joint effort with other institutions. And I can point out this is a collaboration between my lab, Joana Almaza's lab, and with Vanderbilt, with Diane Saunders, Marcela Brizola, and Al Powers, and also Rita Bottino, who is mainly involved in isolating islets from young donors. So one of the interesting aspects about looking at the maturation of human eyelid function in the first decade of life is, okay, first of all, we know very little about it. But second, it's also an age where there is an increase in the incidence of when the antigenicity when the antigens to diabetes appear, there is a high probability that these young people will become diabetic. So this is a stage that is critical that we should take a look at. So first, some anatomical studies that Marcela Brisova performed, and you can see that already at birth, there are islets that contain insulin and glucagon. There are a lot of small islets, and this over time changes to the adult composition, as you can see here. But what is striking is that there are a lot of insulin positive alpha beta cells early on. And the question that we had is, can we now look at the functional maturation of these cells? So what happens over time with insulin secretion and with the responses of beta cells and the different cells in the islets? And this, we performed that in parallel. We were able to record from isolated islets as well as from living slices. And you can see now recordings from adults in gray and from donors that are in childhood, which is between 2 and 10 years. And the pattern of secretion is very similar. You can see a first phase and maybe a second phase, and then also a response to KCl depolarization. What is different maybe is that the baseline is a little bit higher in the adult than in children. And this is very similar between the living slices and the isolated islets. So the pattern is very similar. And this is now glucagon secretion. It looks like glucagon secretion is already mature in childhood, as well as insulin secretion. But it was very difficult for Rita Bottino to isolate islets from younger donors. And there, the slice technique proved to be extremely useful, and you can see it here. So here on the right, you can see the comparison of insulin secretion of children, and on the left are the recordings that we obtained from younger donors. In the neonates, which is between birth and 3 months, there is very, very little insulin secretion, and the baseline insulin secretion is also very low. In infancy, which is from 3 months to 24 months, you can see that the baseline increases of insulin secretion, and there is sort of a response to glucose, and then a response also to KCl depolarization. But it's still very far away from the adult. So these data indicate that this is a process that takes years. It takes years for the islets, although they have already insulin, it takes years for the islets to fully respond, or for the beta cells to fully respond to glucose. We can in parallel also perform calcium imaging experiments. So again, using a calcium indicator that is loaded in all the cells. So here we cannot know which cells are responding. We can do some immunostaining after the recordings, but in this case, we didn't perform it. But what is clear is that if you go from neonatal to adult, that the incidence of the responses increases progressively. And this is very similar to what I showed you before with insulin secretion. By the way, all the cells that are included here are also cells that respond to KCl depolarization. So we make sure that these cells are alive. So this is now we are going to look more in detail into the mechanisms of what leads to glucose responsiveness during these first 2 years of life. A third example, and I think that Stephan already mentioned that, is that we can look at other cells other than endocrine cells if we use slices. And we are particularly interested in the eyelid vasculature as well as the local immune cells. And the person who is mostly involved with the human eyelid vasculature is Rona Massa in her lab with Luciana Mateus Gonçalves. They are looking at the physiology of the human eyelid vasculature as well as in adults as well as during development. And we have been interested in looking at the local immune cells that reside within the eyelid. And I will show you what we have done so far. Briefly, this is briefly a reminder of how the eyelid vasculature looks like. It's composed of capillaries, but the capillaries are not passive tubes. They are surrounded by pericontractile pericytes, shown here in green. And the number of these pericytes is much higher within the eyelid than outside the eyelids. So they perform very important functions, and some of the work has been already published by Joana Omasa and Luciana. I'm going to focus on the function of these blood vessels. And this is something that can be performed, surprisingly, this can be performed in slices. Of course, there is no blood flow there, but what this slide is illustrating is that you can measure changes in capillary diameter within this, with the eyelid in a slice. This is an example. So Luciana and Joanna, they are able to label the blood vessels with a marker, with a lectin marker. So you can see the individual capillaries here in the eyelid. What is nice is also they can look at the feeding arterioles or arterioles that are close to the eyelid. And then you can sort of guess there is a lumen here, and in this case, in the case of the arteriole, you can see a lumen too. If you now apply drugs, stimulants to these slices, you can see that this is over time here, that the arterioles as well as the capillaries, they seem to constrict. And this is quantifiable, and those changes are reversible. You can apply many different drugs to the same blood vessels and look how they respond to different substances. So I just wanted to show you, and I'm not going to go into the details of the quantification, but I wanted to show you that you can get quantifiable data. So this is an example. So they are now also involved in looking, I mean, this is another example, looking at the calcium responses in these blood vessels. So this is a movie. I don't know if it's going to play well, and it's not playing at all. Okay, I cannot. So basically, I will go to the next slide just to show you the results that you can get. But Joanna and Luciana, they are able to identify the pericytes within a living slice and look then at the calcium responses within these identified pericytes. And this is shown in the next slide. So they are looking at the calcium responses to noradrenaline, which is a stimulus that normally stimulates pericytes elsewhere. They are showing that noradrenaline is stimulating pericytes within the eyelids and in pancreatic slices, and they can conduct a lot of pharmacological studies on these slices. They are part of our studies related to the development of the human eyelid. And what you can see here in these preliminary data that are unpublished is that the responsiveness to noradrenaline develops with age. Early on, the pericytes, there are a lot of pericytes in the eyelids already. I didn't show it to you, but these pericytes don't seem to care about noradrenaline. And it's only after 2 years that the pericytes in the human eyelids start responding to noradrenaline. And what is interesting is that this parallels the invasion of the eyelid with the sympathetic axons that normally provide the noradrenaline to these pericytes. Another example now, this is work that was performed by Jonathan White when he was doing his PhD with us, is that you can look at the resident immune cells. And what you see on the left is an image. This is a living slice, labeled, loaded with the calcium indicator in green. You can see the endocrine cell compartment because it has this very strong backscatter that you can detect with a microscope. So this is in the middle here. And then in addition, we use antibodies that people use in flow cytometry to label different immune cells. And this is shown here in different colors. So CD14 and CD45. CD45 is a general leukocyte marker. You can see here sort of cells that look like T cells. But in addition, we have some cells residing inside the eyelid that you see here on the right in B. So these are two cells that we believe are resident macrophages because they express or they are labeled for CD14 and CD45. And you can see them here closely associated with the blood vessels. But this is in vivo. So now that we know which cells we're dealing with, we can now perform functional studies on these cells because we have loaded these slices with a calcium indicator. And this is shown here. Again, we can expose the slices to several stimuli. And in most of the cases, we start with changes in glucose concentration. And you can see that these are responses now from macrophages, from the resident macrophages in human slices. You can see that the human macrophages here, they seem to respond to a change in glucose, but it's probably not direct, but mediated by substances coming from beta cells, because if we block beta cell activity with calcium channel blockers, these responses are gone. These macrophages, we identify them also because they strongly respond to ATP here in this case and at the end. Again, this is showing you the power of the technique. We can now also look at other immune cells. So we have a study going on, not with human slices, but we have a study with using NOD mice, where we're looking at the immune cell infiltration during the progression of diabetes. And we expect to obtain some valuable information that you cannot get otherwise. I think that it's very important to look at the immune cells locally in situ when they are attacking the eyelid. And I think that I will hand it over now back to Stephan. And let me see.
Thanks, Alejo. So, I mean, Alejo has been showing a lot of ways that we can look at, and this is definitely not the limit that we have reached yet. We haven't showed many studies that are ongoing with other people that are getting slices now within the slice group of endpoint. We think the publications that have been going out by various groups have basically shown us that it is possible to use human pancreas slices. So, but of course, at the end, also in light of the upcoming discussion, I would like to put a little bit into context, where are we and what can we do with slices? How do we have to see this relatively new platform, and when we look at human eyelid cell physiology? So, and that's why we have this title with Bridging the Gap, because just from a physiological point of view, if we look at the tissues, the slices are in between what we were using up to date in terms of isolated islets and going to the systemic metabolism. Of course, the gap to the systemic metabolism is far bigger than the gap going to the isolated islets, but we think that this is a technology that goes the next step. So, and that's why just from a physiological point of view, from a morphological point of view, we're going a step closer towards the systemic environment and to learn about pancreas physiology within the human body. I think another way to show this is when you look more in an experimental context, when you look at the resemblance of the systemic system, so the in vivo conditioning condition, the resolution you can get with your technology, and also the throughput. And again, we believe that the tissue slices are somewhere sitting here in between the isolated islets and the in vivo system. So it doesn't, and I think this is a very important point that I want to stress, and Alejo also stressed it. It's not going to replace isolated islets, as some people think we are trying to take over now everything and replace isolated islets. Not at all. We think it's a complementary approach. We think that islets have certain strengths and slices have certain strengths. And we believe that adding the tissue slices now to this will help us all to get closer to a solution of pancreas physiology and pathophysiology in the context of diabetes. So that's why at the end, I would like to put up again that this has potential, but also the limitations. We've been talking about the potential that it has because of the type of preparation it is. It doesn't have enzymes. It doesn't have centrifugation steps. We know from a number of studies now that the cells in slices are better rested than isolated islets after the isolation. And of course, the intact tissue environment gives a lot of possibilities that what we can study either individually or in context in its interaction with the endocrine cells of the islets. We've been talking about how important it is in the immune attack in type 1 diabetes that you can now look at influences that come from outside the eyelid in this intact tissue, and that you can also study come from structurally compromised eyelid. And for one basic question of mass versus function, I think it's also nice, but not only for that, to have a possibility to do function and morphology on the same tissue in right away within the same experiment. There's a lot of more potential. We haven't even touched of all the immune cell studies that are going on with T cells already in NPOD samples. But I also want to point out the limitations. It is regionally restricted. So we take a piece of pancreas and we can say what is going on in that region. We not necessarily know what's going on in other regions. We can take slices from more regions, but of course we can also use that to our advantage and get an idea about how the pancreas differs in different regions. Slices are more delicate in handling than other cells, than other isolated islets. You have to get used to it. And but if you do, it's actually quite easy once you get used to it. So you cannot be afraid of using slices. It's a very nice approach to use to. Of course, you have different cell types, meaning you have exocrine cells that secrete enzymes that you have to take care of when you work with slices. You have a limited throughput in number of islets in comparison to isolated islets. That is something that is limited by the technology, and certain manipulations are far more difficult, like transfections of tissue is more difficult in a slice than it is in an isolated islet. Therefore, also some omics might be reduced. And as I've been pointed out several times now, that's why I put it here, we also have a restriction in islet size, because depending how thick your slice is, that's how big your maximum eyelid size will be. And this has been told to me several times by reviewers now. So we are aware of this fact. This is something that we know, but if you're in a human eyelid with 120 micrometers thickness, I believe this is something that, that we can deal with, but that we have to be aware of. So again, it is not about that this is the holy grail now and will replace all other applications, but it's rather complementary. At the end, I would like to, in one slide, catch up on a discussion that happened on the last HERN meeting, the last in-present HERN meeting, where we were sitting in a working group and discussing if we're using isolated islets and human tissue slices, what donors, what sources do we use? And the discussion was going into a direction that one is bad and one is good. And I would like to point out that there have been different ways to get human tissue. We know about the DIVID study and the biopsies that have been done. We know that surgical resections of pancreatectomized patients can give us tissue. And of course, organ donors that now are coming via NPOD, but also via different other networks. And all of these have advantages and limitations. And this is, again, this is non-exclusive. This doesn't mean this is all. There are different advantages, different limitations to the different sources that we have. And the discussion at HERN back at the time was, is any of these really the normal human pancreas? And probably it's not. But I think with the technologies that we have, the sources that we have, we're getting very, very close there, and we use all technologies and all sources that are reasonable to use to try to get closer to the physiology and pathophysiology. So with that, I would like to end with the take-home message, and that is that human pancreas tissue slices are a complementary approach for the study of human eyelid cell biology. They provide a tissue-based platform to help bridge the gap between cellular and systemic methodologies. And I hope that is what you take with you And I cannot stop without thanking a lot of people here. This is probably, I hope we don't miss anybody. These are people we're collaborating with on the human tissue slices, people that help develop the method and now make it available to the entire community. And of course, we thank the tissue and organ donors and their family and the various funding agencies. Thank you.
Thank you very much, Stephan and Alejandro, for that wonderful talk and giving us a really great number of examples and talking about this. We do have a number of questions, and what I'd like to do is I'd like to alternate between questions that people are asking that can ask live and the questions that have already been asked in the chat. So what I'll do is I'll start with the chat question, but if you'd like to ask your question to either of the speakers, please unmute yourself. I'll be able to see that you've unmuted yourself, and I'll call on you to ask the question. First question is from Sasenka Ramahadham, and she asks, what is the macrophage response being measured? Alejandro, I think that one is for you.
Yeah, so we measure the macrophage response using a calcium indicator. So what you see is a calcium signal produced by the cell.
Okay, thank you very much. Now I'm going to go to a live question from Francois Bout. Would you please go ahead and ask your question?
Hi, I think my question would also be for Alejandro. In your example one, you showed the calcium response in a really nice way. I haven't seen this published yet, so I hope it's already published. Otherwise, I need to update my literature a bit. But for both the diabetic donor as for the non-diabetic donor, there seemed to be very little response in the 17 millimolars of glucose, because you could see, I don't know if you selected these cells specifically to more likely be alpha cells. You see what I'm, yeah.
Yes, yes, you are totally right. Since we don't know which cells we're looking at, and in the case of the non-diabetic donors, we use adrenaline as a marker. So these are pre-selected cells.
Okay.
And,
okay.
Yeah.
Okay, cool. And do you select them by a region of interest after using the adrenaline to select?
And then we have a region of interest that is automatically detecting the cells that respond.
Okay, great. Thank you.
Wonderful. Thank you. Sasenka, sorry about that. Okay, the next question, we'll go back to the chat, is from Les Satin. And I believe this question is for you, Stephan. And it was, you were addressing the limitations. You have a smile on your face. I guess you know the answer. And that is regarding viability with shipping, and how can you improve that?
Well, actually, with the shipping, I think that's rather a question for Alejandro, because he gets slices shipped. So we are not getting, we tried to get slices shipped from inputs from the US to Germany, but that didn't work out. So now I think Alejandro knows a better answer to this.
Yeah, I think we have been looking for shipping buffers. And I think that after many years working together with Gainesville, we have sort of optimized the conditions for shipping. And I have to tell you that the slices that arrive from Gainesville, you may be surprised, it looks small in the atlas and the map, but it's actually, it takes also like 8 hours to get to Miami from Gainesville. And we get a very, very high quality of slices. And I think that within the US, if you would get the slices within the US, I think that the quality would be very, very good.
So Les, if you've had a different experience, please share that with us. But I'm going to go ahead and go to the next question, live question, and that is from Bart Roop. Bart, can you go ahead and ask your question?
Absolutely. So first, thanks to Stephan, Alejandro. Stephan, I always appreciate your candid presentation and the do's and don't do's. And the one thing that you may want to discuss a little bit is the potential impact of all the proteases, the SNR contribution that could induce confounders. But I was very interested in the data of Alejandro on the glucagon, where you showed that 2 of the type 1 slices looked pretty much like the non-diabetic and 2 did not. And that brings in Stephan's concern about the lobularity that we have in the disease. And I wonder to what extent you have already explored whether one and the same diabetic donor could have apparently normal islets versus affected islets and so forth. So to what extent your sampling could basically impact your conclusion of what is diabetic and what actually is maybe not diabetic. Or
Stephan, you want to go? Or I mean, I can only tell you that we can actually see the heterogeneity of the immune cell attack more so in, I'm sorry to say that, but in NOD mice. But we are able to look at islets, and we know the degree of infiltration of the individual islets. So one project that we have is to look at the function of the beta cells and other cells within infiltrated islets versus non-infiltrated islets in the same, even in the same slices.
Yeah, but the curiosity is that some of the MPOT studies using mass cytometry, a tissue side of basically, that what looks like a normal uninfiltrated islet in a diabetic is still not normal, and the slices will be a fantastic way of addressing this. So there is stuff that we don't see by infiltration because that malfunction may precede the infiltration. So I am very curious to what extent we can have a proper representation of a graft, and maybe it's a rhetoric question, but with a lobularity that will be pretty challenging, right? So whatever we, it's an anecdote in the end of the day. Is that fair to say, or am I too skeptic?
Stephan, I think you have a project addressing lobularity, no?
Yes, so it's absolutely right, Mark. We are in our restriction to a certain area. We are always depending on where we end up getting the slice from, and that's why we have one project where we try to go into different areas and get slices from different areas, and then compare those. And what has helped us, for example, in our first study, where we looked at the type 1 cases, is actually to go then to the endpoint database and look at the histology they did to see how much did our slices actually reflect what they saw in their histology. So I think this is always something that you will need to do. But we're trying to address this point by looking at slices from different areas and trying to see how they differ. And we actually, as you say, we expect that there will be huge differences within the same pancreas.
Thank you. That sounds exciting. Thanks very much.
Thank you very much. Okay, we'll go back to the chat now. And this is a question from Ahmed Ghazali. And the question is, and I think it's to you, Stephan, could the limitations of tissue slices be overcome by organoids? You probably have received this question before.
Well, once the organoids are that good that they resemble a human pancreas, I think the organoids can do that. I mean, I don't claim to be on the latest data what organoids is, but the organoids I know are usually limited to a certain cell type or to maybe 2 different cell types. And so I'm not sure at this stage. I don't think organoids can replace the slices. But again, I mean, we will develop new technologies. The slices are a new technology. The organoids will develop, and maybe they will be the next step that we can go in addition to the slices. And so in the future, yes, potentially. But from what I know so far, the organoids are not there yet.
Thank you, Stephan. Bruce, thank you for your patience. Please go ahead and ask your question.
Yeah, thank you, John, and thanks both Alejandro and Stephan. Really beautiful work. 2 quick questions, if I may. One is just for those of us who are trying to set this up in our labs. We've ordered the Vibratome that we'll dedicate. We've got an islet parafusion system, but I imagine there's some nuances to all the connectors and things like that. So, I mean, is there a resource, or, you know, as we get into the nitty-gritty of that, how might we interact with you in terms of setting that up?
Yeah, so this is a question that is very timely because Julia Panzer in the lab is working together with the company that produces the perfusion machine with BioRep, and they are working on a setup that is specific for slices. So far we have been using imaging chambers and putting the slices in the imaging chambers and attaching it to the perfusion machine. But now we are working on producing something that can be used by the whole community, and it will be coming soon.
So it would be all right if someone in our group reached out to you and you can put us in touch with a technical person.
Yes, yes, yes. And by the way, feel free to send people to the lab, and we're happy to teach everybody the technique. Yeah.
Great. And then the more scientific question was, I guess, related to what Bart was saying is, you know, I'm trying to envision this is a parafusion, not a perfusion, right? So all of the endocrine and exocrine secretion gets kind of mixed together and collected. So that does create one issue around proteases, as Bart said, like maybe the prohormones and things get degraded in some of the more sensitive hormones. And then I guess you wouldn't expect kind of intra-eyelid endocrine or, you know, eyelid exocrine axis type interactions to be intact in this. I'm just trying to envision it. That's sort of how you see it. It's in a parafusion, it's a bath, and then everything gets secreted together, not into the, you know, blood vessels or into the ducts as you would in an intact pancreas. Is that right?
Yes, that's right. So we try to keep the proteases in check by using protease inhibitors in the solution. But of course you have all the enzymes, and it's, as you said, it's a parafusion, so everything will be mixed. It's in that case, it's quite similar to the isolated islets in terms of a parafusion. Also, when you talk about paracrine interaction of cells.
Yeah, I guess I was wondering if it, you know, there was old data in perfused human pancreas. People don't really do that anymore, but if there were any differences that you might predict from a perfusion versus a parafusion. Thank you so much. Great data.
Thank you, Bruce. I'm going to go back to the chat. There's a second question from Sasenka, and that question is, how do you normalize functional responses with respect to the number of islets in a section, or maybe the sectioning through the islets, cutting through the islets?
So we can measure hormone content after the experiments. So in the first slides that I showed you about glucagon secretion and insulin secretion from the human slices, you saw that they were normalized to hormone content. So you can measure the hormone content after the experiment. That's
great. Thank you, Alejandro. Okay. I don't see anybody else with their hand raised or unmuted. No, I do see one. Okay. Francois, would you like to ask another question?
Yes, if it's possible. Thank you.
Yes.
I was just wondering on a on a technical setup, whether you, because I've seen in some of the articles that you do an immunostating, but that's of course after fixation. I was wondering if you have tried anything on the pancreatic coops with maybe cell sort antibodies, live cell antibodies, to identify the cells while doing the calcium imaging, or no?
Yeah, this is a great suggestion, and we have not been very successful for the endocrine cells, to be honest. So we are very good at identifying vascular cells and immune cells, but we are using some of the antibodies that also Marcela Brisova has used in Vanderbilt, that were produced by Matthias Grompe, I think so. And they were trying those in vivo, but so far the signal is very, very small.
Yeah, I mean, because I know that we've tried the technique a couple of times for both mouse and human, but it's just to get through to the tissue, it's just so thick. So if you have any suggestions, I mean, but I'm taking up on the suggestion to get in contact, so I sure will. Thank you.
Okay. Thank you. This has been a very enjoyable time, guys. I mean, we've had a lot of good discussion, and I've enjoyed it so much. There's been a number of thank yous in the chat from Martha Campbell Thompson, Amelia Linneman, Les Satin, and of course, I'd like to again thank you for the time in presenting this, pulling this data together, answering the questions, and sharing with the community the latest work. So thank you, everyone, and we'll make an announcement soon as to the next talk. Bye-bye.